Okay, we'll go ahead and get started here. And let's see, you've got 2, 4, 6. So 3, and you all have your book. I would suggest that you grab one and open up to page 600 or 600. Okay. We don't spend a lot of time on these. But then this is not a typical year either. It's one of those aspects to where kind of like reshaping your skeleton. And if you've had anatomy and physiology, is your skeleton reshaped? It is. That is correct. Because what happens is probably the best way to describe that is when you put stresses and strains on your body, just think of, what is it, Disney's, is it Wall-E? Is that something along those lines where it's a civilization where they're riding, or they just ride in, what would you call them, hover seats? Okay. The cartoon, of course, Wall-E. And one of the aspects to where I think as that movie goes on, they're conditioned to believe that they have to have that hover vehicle. Would that be correct? I've only seen it once, and that was years ago. But that's just what seems to resonate in my mind. But what happens is when instances like that happen, when we don't put stresses and strains on our bodies, your skeleton can start to weaken, just like muscles do, okay? Because it's that, what would you call it, an old adage or a proverb where you say, if you don't use something, what happens? Like with muscles is the best example. Yeah. So you're losing it, right? Isn't that how it goes? You don't use it, you lose it. Okay? And hopefully for our purposes, it's just like perhaps riding a bike. Should things look familiar from the first semester? Well, I would like to think so. It might look familiar, but it's perhaps just like getting back on that bicycle and pedaling off into wherever it is around campus, perhaps. Something to that effect. But what happens is the skeleton will reshape itself where it will take, you could say, I wouldn't say sections, but digest portions of your bones where they can be shaved off, okay? And then build that tissue back up where it needs to be. So then what happens is the analogy with this is we are cutting materials out where we can afford to, and then put that material back in where we think we need to, and that will be with the lipids. Because one of the things that I think has resonated over the years from students in the past is, are they thankful for this class or taking it? Yes, I believe so. But that only will last for so long because we can only cover so much material, and let's say so far in depth as far as for what our purposes are. Then once they get into the lipids with perhaps fat-soluble vitamins and water-soluble vitamins, that's where things maybe start to get a little difficult. And that's what will be next after this portion. So what we see on page 600 is a list of what? Okay, so then what is this chapter entitled then? Okay, so how do those two, how do they go hand in hand? Do we know? Well, when you say that, you got to be careful. We could say a leukocyte. Well, isn't that a blood cell? Well, yes, it is. It's a white blood cell, but it's a blood cell nonetheless. But when you look at this, amino acids are the components that make proteins, okay? And where this, we could say, maybe finally comes full circle, okay? Maybe this brings back good memories, maybe not. We could say things like this. So how or why does that look familiar? Yeah, it is DNA. And what we had discussed last chapter was, perhaps you could say, well, I almost did that wrong. Okay, well, I did do that wrong. Oh. What do you think? CH2OH or OH there? So we have one going either way.
It's a OH. It depends on if it's a ribose. It's ribose.
Yes, it's ribose, so therefore that is an aldose sugar.
That's not ribose, Ben. Yeah, it is. Oh, yeah, you're right.
Okay, then it could be something even more specific. Okay, these are all in here. Then what is this now? Because that is a real molecule. That's deoxyribose. So we're putting some of these concepts together where we got this 5-carbon sugar that is missing an oxygen here, and that's why we call it deoxyribose. And of course they are nucleic acids. Now, so 3, 6, 7, 8, 9. Okay. Does anyone possibly remember why we had 7 up there? So I put 2 more on there to make 9. Because 9 is a factor of what? It is. So then what would happen is in your biology class, and you will be doing this in your freshman level biology class as well. Okay, so what's going on here? Here. Yeah, that's a T, sorry. Okay, so, and good question. How did you know that? Because someone said the phrase that we used. Well, true. You have to know where you're at to know if you're coming or going, right? So you have to know where you're at to know if you're coming or going. Okay? I don't know if that helps or not. But then this next part, okay, here's your DNA molecule that is double-stranded. And then what happens when you see something like that? This should tell you something really, really specific. Because if this is double-stranded and this is single-stranded, what's a nucleic acid that's single-stranded? Come on, I got the boss in here.
Don't be shy. It's okay to show them that you know something.
Mm-hmm.
Ben's kind of got my look down. Kind of look, and then you just
Okay.
kind of onto it, and then you kind of flip the page. There, I'm not even close.
Okay.
No, that just kind of buys a little time.
Okay.
Did Schaefer's ride today?
Yes, they did. I'm going to hit pause for just a moment. Again, DNA is double-stranded. What must be single-stranded?
RNA?
RNA is single-stranded. That's right.
See?
So then when you made these matched up as codons, these codons then would match up with what we call anticodons. Okay? because thymine replaces uracil for some reason. Or, excuse me, uracil replaces thymine. Okay? It is, but the important part about this is when you look, okay? Now, this isn't a, what do I want to say, part of the nucleotides, but where do you see this somewhere on 600 or 601? Look for MET or M-E-T, because it stands for something. Okay? So it's what we call methionine. Okay? Methionine is a very specific amino acid. Okay? This meant nothing to you when you're sophomores, nor should it have been. But now that it's come full circle, this is coding for that specific amino acid, methionine. And if we're really good, AUG was what we call the starter codon for the process of transcription. So what happens is RNA polymerase is looking for this particular amino acid so it can start reading the DNA molecule, okay? Because this is coding for that methionine. Then why that is important is now you can get into your specific amino acids, which means, okay, how many letters are in the alphabet? So when we look at the different combinations those 26 letters can make, just pick up a dictionary, okay? And of course, how many of you even use a dictionary? Well, yeah, everything's on our phone. Okay, I'm not saying that's good, or I make it sound like that's terrible, but yeah, everything's at your fingertips. But remember, you can still use one. But you got to remember, your phone doesn't choose words for you. The computer does not choose words for you. It's one of those aspects. They can't write for you. You still have to be able to write and write well. And I think you would agree with that. So that's sometimes why when you're taking those higher level science classes, what do we tell you? You need to be able to write and write well. Okay? So this is where all of these particular amino acids have something in common. Okay? So getting back to, you could say, the English language with 26 letters, the different combinations of those letters makes up such a big combination of so many different words with many different meanings. So with 20 different amino acids, they're the building blocks of proteins. It's no surprise that there's going to be such a wide array of proteins that have very specific functions. We know that they're enzymes. So catalytic proteins, they're building and breaking things down in your metabolic activity. Transport proteins. These proteins are in your cellular membranes that open the gates and say, okay, let molecules in that are too large to pass passively by this process that starts with a D. No, there's a couple F's in it too. Diffusion, okay? Well, these transport proteins will allow materials in that are just too big. It says open the gate, okay? Let's let our polysaccharides through. What might be a polysaccharide that we talked about last chapter? That's a disaccharide. But that still would have a hard problem getting through as well. It's just too big. So is glucose, which is a monosaccharide, but something that's even bigger than both of those. Come on, pasta people. Okay? That's in plants, but that would be one of them. And then starch is your other one. Okay? So these transport proteins have to be able to function to open up, let materials in through your cells, or into your cells. Okay? Regular proteins, okay, controls many different types of your metabolism. Okay? There's not going to be one set of protein or amino acids that build a particular type of protein is going to have the same general function, okay? And we know that because I'm picking on A and P students again. How do proteins get broke down in your small intestine? By enzymes. But can you name possibly some of the enzymes? One starts with a P. It's kind of like a soft drink. You have Coke and you've got Pepsi. Pepsin. That is an enzyme that breaks down proteins. Okay? And then where do you find that pepsin? Well, it's made in your stomach, okay, before it moves on to the small intestine. But that's not the only source. So my point is, you're not always going to have the same type of enzyme always breaking down different types of molecules. There's bile that breaks down fats. You have pancreatic amylase, pancreatic lipase that breaks down those substances. So when someone has problems with something like Crohn's disorder, okay, they can't digest things. So if you give your dog a pan of chocolate, they're gonna devour that like nothing else. But you're shaking your head no. Yeah, that's not a good idea. Sometimes it happens. Are we? Right, so it travels right through them. So you might have a mess to clean up when you go home because they don't have the enzymes needed to do that. And sometimes that happens with people. Okay, so enough of that. All of these have the same base structure, okay? Because when we look at something like this, okay, to learn these, you'll see that you have spots in there that has places you can, that's some of the more common ones that we want you to know. There's quite a few of these amino acids we want you to be able to draw. So whether it's on paper, you know, I've made these note cards a while ago, although silly me, I've got the names on the front. That probably doesn't help. But if you really, really want to place something into your long-term memory, I think you would agree it's one of those aspects. You write it down on a note card, maybe not put the name on it like I did, maybe put it on the back, but the more you write them out, the better it's going to be into your long-term memory.
So
okay.
We have to know the structural formula?
Not all of them. That would be way too many. I'll give you a list, okay? But this is how you draw amino acids, okay? I mean, what you see on the top of page 600, okay, it's very similar to that. Now, whether you do this is how I draw. I just always, because that's how I learned it, okay? So let's say, for instance, when you're done graduating from SDSU, you're going to start working, well, being on the family farm. How many, can you think of something along the lines of, well, this is the way I'm going to approach planting or harvesting, because that's how Dad did it. Can you think of an example of anything that I'm going to approach doing it this way because that's how Dad did it? But does that make sense? Yeah. Okay? When I'm explaining these, I do it in this particular way because that's how I learned it. So then try to pass this information off to yourself. Then what you see on the very top, okay, not with your particular amino acids, okay, it's this guy right here, okay? Does everyone see what I'm talking about? Okay? So that's what I mean. They have the same base structure. Okay, and then this is hooking to a carbon, okay, and then another carbon here. Is that showing up on? Yeah, it shows up just fine. Okay, then we see that. How do we designate this as an acid. Okay, double bonded oxygen and, okay. Now, it could be such an aspect. Would you see something like this? You could, but we're not going to do it that way, okay? Because when you show the dehydration and start linking these together, it just makes more sense to see it in this fashion, how we've been doing it all along. And then, okay, we're satisfying the octet rule each and every time we do this. It's just one of them things to where when you're a freshman and then a junior and then, well, how are we going to apply this again? And what this illustrates is what I don't like, okay? They have this, they have an hydrogen here, but I prefer to put it up here, okay? And then we're flipping this over, okay? Because what does that mean? What is R? Can be. And right, it's where these amino acids start to differentiate, okay? All right, so nitrogen is in column 5. So how many electrons is that? Okay, so if we put an electron here, that doesn't work. There, there, and there, okay? Okay, that's only 3 of nitrogen's 5 electrons, okay? And if you remember, how did you illustrate this covalent bond? It's a triple bond, okay? Same thing, okay? But one of the things we said, don't do this. Yes, that satisfies the octet rule, but these electrons are unshared. So do you remember how did we want you to illustrate that? Yes, so when you say on the outside and put them together, we want to say keep your unshared pairs of electrons together. Okay, so you correctly said this nitrogen has 3 X's, but how many more should it have? Just like what we see here, keep your unshared pairs of electrons together. So then there must be a bond here, and what does it say that belongs next to that hydrogen? What does it say that belongs next to this nitrogen? 2 hydrogens, okay? So whether you want to put hydrogens on there, which is not necessary, okay? You can just leave them like that, okay? And then, of course, with this carbon, okay, there's a dot, there's a dot, there's a dot, there's a dot, okay? So carbon will have satisfied its octet rule, and the same thing here. It's one of those things to where it's something that you learned a long time ago. It's just we're starting to have some of them concepts come back again. I put them as dots. I just use X's in this example, okay? Because like, for instance, when I would prefer you did, because like I said, when I practice these, you can see that there is a couple of dots there. So when I'm practicing these, that's what that looks like, okay? Then, as we look at this, okay, we can take that H off because we know that that's a hydrogen. Have we illustrated any amino acids yet? Yes. Sorry. It was a trap, and it was a trick question. We've got an amino acid right there. Okay, thank you. If you look on 600, what amino acid do you think this might be? Why are you right? Yeah, it's just a hydrogen. So what this is illustrating on 600, okay, they put their R groups up here, which is all these different structural changes between these amino acids. To me, it makes more sense putting the R group down here. We know that these are hydrogens, okay? So why do we call this an amino acid? We've already illustrated anything on this side of that nitrogen. Why do we know that that's an acid? Because we have COOH. This, from way long ago, back in August and September, we only mentioned this once, and we said the next time you see this, you'll probably like learning this, because then we know we're in the spring semester and school's starting to wind down. Although that probably doesn't look like it when we peer out the window, but nonetheless, we've got an NH2 group, which means that is an amine or an amino group. That's how it gets its name. We've got an amino group here and an acid group here. That is your first amino acid. Okay? Then, now they start to change. This one starts with an A. If you put a methyl group there, that is alanine. Okay? Then,
You know that one, too?
yes, for sure the first 7. I mean,
Oh, those ones just aren't on our paper. Alanine.
okay. Well, that's why we're going
Should we
over.
write that one
I would write that down. Okay. Then, okay. What is an isopropyl group? Now, don't look at page 600, but what is the structure for an isopropyl group? Exactly. Okay. So when you put these in a row, okay, if you have an isopropyl group on here, okay, perhaps one of the things you can use to help yourself out, okay, to me, there's really nothing wrong with rewriting it like this. If that helps you out. Because what does this look like? An upside down what? So therefore, that must be the amino acid what? Valine. Okay?
So, are they going to be written out like that a lot, or are they going to be written out like molecular formula?
Okay, I understand your question. I just don't really, the best way that I can answer that would say, draw the amino acid methionine, draw the amino acid serine, threonine, okay? Things like that. So you know all of these are going to be the exact same that's in this color. It's at this spot where these are different.
Yeah, that's what I mean. It's like, would we, like, if we're given to name them, is it going to be drawn like that, or is it going to be drawn?
Oh, is going to be given the name like this, M-E-T, okay? Draw methionine.
She's asking if, like, we're gonna have something where we have a worksheet where we have to name them and they're drawn out.
Like that?
No, you don't name them. You always draw them out.
Oh,
Yes. Okay.
But you could draw them either way.
What do you mean? Okay, sorry.
You could put the CH3 parentheses 2.
Yes, you could do that too. Yes, yes. Sorry. Okay. Then, since we talked about this, let's see how my memory is working. I think we go in a series of 3. Then there's an S. Is that correct? For methionine. Methionine is, oh darn it, I put it in the wrong. It's after the second one. Oh, I put one too many on there. Okay. Now, this probably looks difficult because you just haven't spent enough time on this yet, okay? So, if you want to take, there might be some pencil marks in your book already. So, the first 7, which is glycine, alanine, valine, leucine, isoleucine, phenylalanine, methionine. Then go over to the next page, 601. Cysteine, serine, threonine, asparagine, aspartic acid, and glutamic acid. You are responsible for those structures.
Aspartic acid and glutamic acid.
That's correct. Okay, I think that's enough. We'll stop there. We'll catch up to you next time.